Hello everyone, I Assistant Professor Pragati Shete from Department of Microbiology, KG Somaiya College. In this online lecture, we will study about the Japanese encephalitis virus. Okay, in the last online lecture, we have completed till the FMDV virus and the rinderpest virus, right? So let's start with the structural properties and the genomic properties. First, what is the structure? So starting with the family, this virus, Japanese encephalitis virus, will come under the family called as the Flaviviridae. Okay, FVD, which is also denoted as the FVD Flaviviridae, with the genus Flavivirus. Okay, now basically this virus will cause encephalitis, means what? The inflammation, right? And then inflammation of the CNS. Okay, now it is mostly transferred via vectors, the mosquito vectors, and that is why it is also can be called as the mosquito borne encephalitis. Okay, there are various names of this virus, okay, such as the arbovirus B encephalitis, then mosquito borne encephalitis, then brain fever. Okay, as I mentioned, encephalitis means the inflammation of brain, and that is why the virus name can also called as the brain fever. Okay, also it can called as the summer encephalitis. All right, then it mostly transmitted or infected in the animals. Also it can rarely infect in the men's. Okay, it is not in the case that men's will just not get the disease, so men can also get infection, humans can also get this viral infection through the mosquito vectors. Okay, now we will study the genome. The genome will contain single stranded positively sense RNA. Okay, molecular weight of this RNA is 3 into 10 raised to 6 Daltons. Okay, you can see in this picture the genome of the virus is single stranded positively sense RNA. Okay, then the viral structure is the spherical shape, then it is enveloped virus. Okay, important property, it is enveloped virus. Okay, and that is why we will have then enveloped proteins, right, the antigens which will responsible for its replication or for initiating or regulating the pathogenesis, right, in the host body. Then lipoprotein enveloped particles present on the envelope called as the lipoprotein enveloped particles. Okay, being the 40 to 50 nanometers in the diameters. Okay, these are the 3 structural particles and the 7 non-structural particles. Okay, which means we have total 10, right, 10 lipoprotein enveloped proteins. Among them 3 are structural proteins and 7 are the non-structural proteins. Now look into the picture. We can see the outermost lipid envelope, right? On that lipid envelope we have various kind of the lipid proteins. Okay, now inside the lipid envelope there is the protein capsid, blue color one, right, icosahedral protein capsid. Then inside that protein capsid there is the polymerase present. Okay, here is RNA polymerase present with the genome, which will help the virus to polymerization of its genome in the host cell, right, to multiply and to increase in community and to help in the localization of the virus, right. Then we have the genome that is named as the nucleic acid. What is the nucleic acid present? Single stranded positively sense the RNA, ribonucleic acids, right. And you can see there is the spike projections that has been mentioned. So those are the proteins, all right, lipid envelope proteins. These are the properties of the genome and the structural properties of the Japanese encephalitis virus called as the JEV. Okay, now we will see the resistance properties of the Japanese encephalitis virus. Now we have saw the etiology, right? Etiology means what? The structural and the genomic properties. Now we will see the resistance properties. In that, it is affected by the heating at 30 minutes at the 56 degrees Celsius. Okay, and its thermal inactivation point is 40 degrees Celsius. Okay, which means the temperature at which it will get inactivated is the 40 degrees Celsius. All right, now it can also get inactivated with the pH between 1 to 3. Okay, and it is stable in the alkaline environment. Okay, alkaline means what? Between pH 7 to 9. Okay, below 7 it will be the acidic pH, right? That is why in the acidic environment it is not resistant, it is susceptible to the acidic environment, right? It will get inactivated. Then which chemicals or disinfectants we can use to kill this virus or to inactivate this viral activity? So those will include the organic and the lipid solvent, then common detergents, then the phenolic iodophores, then 70% ethanol solution, 3 to 8% formaldehyde, 2% glutaraldehyde, and the 1% sodium hypochlorite. These are the examples of the chemicals or the disinfectants which are affecting the virus activity. Okay, if we use these chemicals or disinfectants, then the virus will get inactivated. Okay, now virus will also is sensitive to the UV light and the gamma radiation. Okay, these are the resistance properties, right? Now we will see the epidemiology of this viral infection. Okay, in that we have 4 types, or we classified the 4 groups in the epidemiological studies, namely geographical distribution. Okay, in this will include which areas are most affected or are most susceptible for the infection. Then what is the host, which are the host, transmission, how the virus will get transmitted, and the morbidity and the mortality rates accordingly. Alright, so now we will start with the geographical distribution. In the geographical distribution, the endemic in the temperature and the tropical regions of the Asia. Okay, which means the Japanese encephalitis virus can cause the endemic diseases, endemic effect in the temperate and the tropical regions of the Asia. Endemic is what? Very limited population will include in the endemic region. After endemic there will be epidemic, and after epidemic there will be pandemic, which is the highest, which include whole world. All right, now it can cause the endemics. Okay, then it has shown reduced prevalence rates in the Japan. Okay, the prevalence rate was reduced due to this virus. Then it has not been occurred in the US till this, and then in India also it has reported the epidemics. Okay, this is the geographical distribution. Means what we studied the different various areas, and in that areas what was the effect of this virus. Okay, now we will study the host, what is the host range for this virus. Now as we mentioned earlier, this virus is mainly for animal. Okay, animals are mostly get affected, although humans will also get affected in some percentage. All right, then starting with the horses. Okay, the host can be the horses, pigs, birds, that is the swines, okay, and the humans. All right, now first, horses. Horses are primary affected domestic animals of the Japanese encephalitis virus. Okay, though essentially a dead end host, other equids, donkeys are also susceptible to this virus. Alright, it's compared to the horses. So who is the primary host for this virus? Horses will be the primary host. Okay, the infection or the most affected host will be the horse. Okay, then next is the pigs. Now pigs will act as the amplifier for the virus. Okay, which means the virus will produce or will multiply, will amplify themselves inside the pig. Okay, now pigs act as the amplifiers, right? Amplifiers of the virus, producing high viremias, which means high concentration of the virus in their blood, and which infect the mosquito vectors. All right, from the pigs the viral will transfers to the mosquito. Okay, if a mosquito bites an pig, infected pig, we cannot call it as the infected, we should call it as the pigs who has the virus in it. Okay, the virus got multiplied inside it, right, inside the pig. Now the mosquito will bite that pig, and therefore the infected blood, which having the high concentration of the virus. What the hell, yaar?